Our general big topic would be evolutionary genetics, and within that we are particularly interested in how new morphologies evolve. So, and our big question is what sorts of changes in the genome, in genes, underlie all the diversity in shape and form that you see among organisms in the world. And we particularly work with corn, or maize, and it has a wild relative called teosinte. And so we address that big question using corn and teosinte as our model system. We look at quite a few different genes and quite a few different traits. And in general, our approach is to start with phenotype. So how does corn look different from teosinte? And so one way they look different is that corn, if you take an ear of corn, the kernels are right there on the outside of the ear. If you take an ear of teosinte, each of the kernels has a hard little casing. Another is the size of the kernels. Teosinte kernels are much smaller than maize kernels. Another is the shattering trait. In teosinte, as in all wild plants, when the kernels get mature, they fall off the plant into the soil so that it can naturally start the next generation. In corn, all the kernels stay firmly attached to the cob, and the cob stays attached to the plant. So there's no shattering or means for natural seed dispersal in corn. And so we would start with that difference in phenotype, or difference in how the plants look, and then we would try to work back and find out what's the gene that controls that difference. In this case, what's the gene that controls the formation of this casing that surrounds each of the teosinte kernels, but is not found on maize kernels. The way we identify the genes is by what we would call genetic mapping, and that is that we kind of search along the chromosomes to find regions of the chromosomes that are associated with the differences in the trait appearance in corn versus teosinte, and we narrow those regions down to a finer and finer scale till ultimately we can get them down to one gene. And actually we can get them even finer than that. We can go within the gene and we could say the reason that corn looks different from teosinte is that because in corn, at this particular amino acid site in the gene, there's a lysine encoded, but in teosinte, at that same amino acid position in the protein, there's an asparagine encoded. So in many cases, we can actually trace the difference in appearance back to a single amino acid change. One of the greatest benefits that comes out of the research is training new scientists. So students and postdocs, graduate students, and undergraduates in my lab, many of them go off to work in the plant breeding industry. And so they may work for Monsanto or Pioneer Hybrid or a vegetable crops breeder or something like that. So one big aspect of it is that they're getting training working on these projects that's applicable to modern plant breeding. Yeah, so what I do when undergraduates do a project in the lab is assign them to one of the graduate students or postdocs to act as their mentor. And then we try to give them a project or we do give them a project where they have some question or hypothesis. They will collect the data, they will analyze the data, and at the end of the semester be able to have an answer to the question or hypothesis that was posed. And then we also like to have the project have a couple different components. One component would be maybe working with the plants and actually measuring phenotypes or traits on the individual plants. But then also another component where they would work with the DNA extracted from the plant and actually look at variation in the genes themselves.
Because teosinte needs a very specific day length in order to flower, so we can't just put it in a field and expect it to flower and produce any seed, because it has to have a specific amount of day, actually it's night, it has to have a certain number of dark. So we grow it in here, and then actually last summer we transplanted some of it once it had been induced to flower. But yeah, we're just growing some